Good evening, Pittsburgh and universe. And this is JC on a bike. My name is JC and this is Journal Club on a bike. I don't have a full battery, so we're going to do an extra short one today, but I hope that my plan to do a short one will coincide well with the short battery. I want to discuss the diversity of the human cortex today, and specifically sort of address the idea that we've been looking for a long time for the physical and structural evidence for why humans are smarter. And one of the things that we often look at and cite for the structure that makes us smarter is the wrinkly part of the brain, the neocortex. And so the idea is that the wrinkly part of the brain has expanded in primates, and that's resulted in our higher intelligence. Extra connectivity, extra circuitry involved, extra circuitry available. And so the idea is that whatever changed in the neocortex underlies our intelligence strength. Now, anatomists have long cited that layer 2-3 of the neocortex is expanded, and that the entire cortex is expanded. It's not so much an increase of cell density, but it's more cortical area, and specific layers have gotten thicker, especially layer 2-3. So the first paper I want to talk about today is by Matthew Larkum's lab in Berlin, and he shows a very unique computational property in the apical tuft of human layer 2-3. We have known that the apical dendrite of pyramidal cells is special for a long time, but I still don't think we have really fully understand the functional relevance in terms of what pyramidal cells can actually do with their super special morphology, this apical dendrite. Previous studies have shown that back-propagating action potentials originating in the soma can go back out into these dendrites and cause plasticity to occur. And some of these dendrites have been shown to have some electroactivity of their own, meaning that they can generate active potentials on their own. Most of these active potentials are still sodium-based, although there are calcium channels in these apical dendrites as well. And what this paper purports to show is that the dendrite of layer 2-3 pyramids in humans is capable of generating calcium-dependent action potentials that are independent of the soma. And historically, we haven't really been thinking too much about dendritic action potentials except for in reference to somatic action potentials, thinking that the idea was that the machinery in the dendrite was just more accurately conducting the somatic action potential back into the dendrites. But there was another paper not too long ago, which before this ride I could not find again. But it was in electric fish. And what it showed was that in the electric sensing organ of the mormarid fish, that the dendrites of these cells were acting as a hidden layer from the perspective of a computational network. And this is cool because computational neuroscience has long understood that, how basic neuronal networks work, and how neuronal networks can be organized into layers, and then those layers learn in between those layers, and And then the more layers you have, the more a network can learn. But what hadn't been shown until this Mormerid paper was that the apical dendrite of pyramidal cells could act as a separate functional compartment. So in this paper then they're showing that in humans this separate compartment also has its own electrotonic potentials that are dependent on calcium. So the electrotonic potentials that occur in these dendrites are capable of firing completely independent of the soma's action potential, which means computationally this cell has two compartments which can in theory work separately from one another. And that same basic idea was shown in this Mormerid paper that I couldn't find for this ride. So the machinery required is present in human layer 2-3 pyramids and not in layer 5. The layer 5 pyramids are also discussed in this paper, but they don't show this independent property. Other people have looked. I think Mark Harnett looked, for example. So the last paper that I want to mention is a paper by a lab called Polleux, P-O-L-L-E-U-X. And this guy, his lab is in New York. And what he has done in a previous paper is expressed a human variant of a GTPase, and the mouse analog is SRGAP2A, and when you double this gene is duplicated in the human genome, and if you express the human version, it suppresses this 2A gene product and instead replaces it with a 2C product. And this 2C product, gene product, is the human version. And so what they've done is created a mouse line where they can express this gene, this human gene, in specific neurons in the cortex, and then using rabies tracing, they can start to measure the connectivity of these neurons and see if the expression of this gene changes their connectivity. And the long and the short of this is that when they express this gene, this human gene, in cortical neurons, what they get is a specific layer 2-3 change. And that is that layer 2-3 has more cortical to cortical connections while preserving the ratio to subcortical connections, to thalamic connections. So with this quantitative rabies, they show that this single gene duplication in humans may be responsible for the thickening and increased connectivity in layer 2-3, which is the reason why Matthew Larkum's lab looked at layer 2-3 pyramids in particular. So it's cool because there's only one gene here that they're talking about, and one gene results in this huge physiological change in this specific circuit, in the specific circuit of the cortex, but it's a generalized circuit throughout the cortex. So I think that's where I'll wrap it here, and I'm going to leave it like that. The two papers I got, I don't know if I can find the Mormerid paper or not. I'm going to try and find it and include it in the links here. But suffice it to say that what they showed in that paper was that what the dendrites can allow to happen is continuous learning, and it's based on the idea that the apical dendrites function as a hidden layer, which previously, at least computationally, we hadn't thought of it that way. And in thinking of it that way, the physiology of this mormorid, the physiology of the learning was able to be explained. So, layer 2-3 pyramids. What are they? What do they do? What are they and what do they do? I think we've been on pyramids for a long time, trying to figure out what pyramids do and how they do it. And what this, apical dendrite was for. Was it really just for the soma to listen to the upper layers, or is there more functionality to it? And again, historically speaking, the backpropagating action potential was the mechanism by which synapses on the apical dendrite were subject to plasticity, to change. But now it could be actually that many or all of these cells are actually performing calculations using this apical dendrite as a separate layer in a computational sense. And that is a great step forward in terms of understanding, or in terms of possibilities, in terms of how the brain works. In light of all of our recent data with correlate this to that and that to the other, this is a very nice result. These are very nice results because they're starting to speak to, how the brain works. And I like papers that speak to how the brain works and add to our understanding of how the brain works. And I think these papers, they single in on or zero in on layer 2-3, but I think they show how, papers can come together to try and collectively push an idea. In this case, trying to understand how the human cortex is different. Keep in mind that we can show you this picture of all the cortical regions in mammals, and you can see that for the most part our assumption holds up right until you go to cetaceans, and then you see that they have even more wrinkles than us and more cortex than us. So the question then becomes, what's the schmo doing? What are they using all that cortex for? And are they as intelligent enough? Because that's the question, right? I mean, if you stop there with the big brain of the human, you can make a really deceptive argument that, it's our wrinkles. But then you show a dolphin brain, and it's, can't be. Because if it is, then we're not the smartest ones on the planet. Or at least we don't have as much machinery relative to other animals as we thought. Thanks for watching. If you like and subscribe. This is JC from Pittsburgh. Love your neighbor and register to vote. Register to vote!